This next gentleman, I'm going to introduce him, although he needs no introduction. And he does have some good points in spite of the fact that he's an Ohio State fan. We have to take into account the scores of papers he's written on lung cancer and, of course, the patents that he holds. So please give a warm welcome to Dr. Eugene Woltering. Eugene, where's the little clicky thing?
As Robert says, I'd like to help you out. Which way did you come in? We're going to talk today about some of the new stuff that's happening. We're going to talk about things that you may not have ever heard of, you may not have seen yet, but you're going to see. I was thinking with Dr. Liu's talk, and he was talking about PRRT as the thing of the future. The first PRRT patient in the United States who the family allows me to talk about was done in 1996 at LSU. So we have the distinction of being the first group to do PRRT in the United States. And the guy who had that therapy, you probably know, being from this area, owned the Tampa Bay Buccaneers. Hugh Culverhouse was the first patient in the United States to have PRRT. And Mr. C, as we all knew him, was truly a pioneer. He was willing to try PRRT when no one had ever done it in the United States before. Eric Krenning had only done 2 cases in Europe, and a whole team of us assembled to do that. Anyhow, what's new in our lab, as you may or may not know, we have 2 full-time PhDs. We have about 6 people that work in the lab. We just expanded to our 6th laboratory. This year, our lab will turn in probably 2 or 3 NIH R01 grants. Of course, we won't get one because of the way the NIH funds things. But at least the research is coming along, and many of the things that you heard Dr. Liu talk about, like you probably missed it, the lutetium going into the cell, were originally described in the New Orleans. Okay. Well, what are genes, and why are these things going to be important? And the answer is genes are made up of DNA. DNA contains the genetic information. It's sort of like a recipe, and everybody's recipe is different. Oatmeal cookies are different than chocolate chip cookies, even though they both have sugar in them. As you may or may not remember from high school biology, DNA is made up of things that are called base pairs. They're adenine, thiazine, cytosine, and guanidine, and they pair up C to G and A to T. These DNAs are then stranded, one from mom, one from dad, into a chromosome, and the gene becomes that sort of coded information that tells the body what's going on. As you can see over here, this is the DNA is the double helix that Watson and Crick talked about originally, and these pairs here are very A to T, C to G kind of things. And there are what are called long arms and short arms of chromosomes, and where your genetic information for MEN1, carcinoid that has a familial thing will be hopefully down to one small area in the chromosome that we'll be able to figure out where it is. As you probably remember from high school biology, or maybe you wish to forget, when the cell starts to divide, all the DNA lines up, all the chromosomes line up. The chromosomes that are like this now make into parallel pairs. One goes with this cell, one goes with this cell, and you create a new cell that way. Mutations are what, up to this point, if you donated your money to genetic research, people were looking for mutations. And what is a mutation? It basically says that here's what the normal gene looks like, you know, A, B, C, D, E, F, G, H, I. And the mutation looks A, B, C, D, E, F, G, H, 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 H. And that's the sort of the idea of a mutation. The trouble is that the information gleaned from a mutational analysis of the entire people in the United States doesn't necessarily help you. It doesn't define whether a drug will work in you or whether you're going to have a good prognosis lesion or you better start packing because God's coming for you. Why are genes so important? They're associated with specific gene states and disease states. If you have MEN1, the menin1 gene is associated with that. What we're looking for in our lab is not a mutation. We're looking at each individual person's tumor that we operate on, and they give us permission to study their tumor. We look for a gene that would, or a set of genes that would code for carcinoid, code for pancreatic neuroendocrine tumors. But we're in my lab, if you look at the almost 170 peer-reviewed papers that come out of the lab in the last 20 years, they've all been about drugs and how drugs control either blood vessel growth or otherwise called angiogenesis, or trying to define how drugs work on the tumor cell. And ultimately, our new focus is, can we find out the genes that predict success or failure with a specific drug pathway? I.e., if you ask me, Dr. Woltering, should I take black raspberry powder, I'll be able to look at you and say, we're going to take a piece of your tumor from the old days. It can be from your original wax block that you had to biopsy 10 years ago, and look and see if you have the genes that are turned on and turned off by black raspberry. And I'll talk a little bit about that today. There are only 28 genes that it looks like, and we just got that paper accepted on what genes are turned on and turned off by the black raspberry powder. So for those of you who don't know what I do and what our lab's done for years, we have a firm belief in that if you look at the progress that we've made against cancer in general, it's been fairly abysmal, except in certain tumors, over the last couple of decades. And the reason is that bacteria and cancer cells are smarter than ever I thought to be, whereas blood vessels are dumber than a bag of rocks. So if I can't fix bacteria, if I can't eat or cure the man-eating bacteria, or we now have in New Orleans a child who just died of an amoeba infection that went up the nose, and in St. Bernard Parish, the water supply for the whole parish is infected with this amoeba, you can drink it and it doesn't hurt you, but if it gets up in your nose, it crawls in your brain and kills you. So bacteria are smart. They learn to mutate. They learn to change their drug resistance patterns overnight. Breast cancer cells, colon cancer cells. Why do we fail? Because those cancer cells are smarter than the doctors who are treating them, and they become resistant to the chemotherapy very, very quickly. Whereas endothelial cells, if you think about hopping on the blood vessel train in your brain or wherever, there's a continuous, if you go long enough, you will go in and out of every blood vessel. Every blood vessel in your body is contiguous with every other one. If you also think about angiogenesis, that in adults, angiogenesis just doesn't happen. You don't grow new blood vessels unless you're a woman having a menstrual period. If you're a guy, once you would go through puberty and hit adulthood, unless you have something pathologic, you don't ever grow a new blood vessel. So that there is stability of what I look at today versus what I look at 10 years from now. Angiogenesis is most of the time very tightly regulated, but it can be unregulated in a number of disease processes. So what I'm talking about today will be carcinoid and cancer, but please feel free if you have diabetes to think about, hey, this may be a way to keep me from going blind, or if you have psoriasis, this may be a way to stop my psoriatic plaques from happening, or my psoriatic arthritis like Phil Mickelson, or tumor growth, etc. So it doesn't happen just in cancer. So what we've been hunting for are the genes that are triggering a resting blood vessel, which in you or me is all the time. All our blood vessels are asleep. And I want to know what it is that turns them on, what wakes them up to start feeding the tumor. Think about it. If you have a tumor, you got to get blood. Why do you got to get blood? Because you got to have oxygen. You got to not only have oxygen, but you have to have sugar. By the way, somebody asked the question about sugar. No, sugar doesn't cause cancer, and yes, you're allowed to have sugar if you're a cancer patient. But anyhow, and you have to have ways to get rid of cellular metabolism products. Sort of like pooping every morning. If you could never poop, you'd blow up, you'd be ultimately FOS. Same kind of thing. And if you think about it, how does a cancer cell escape from that tumor and end up in your liver or your brain or your bone? The answer is it goes into the blood stream or the lymph stream and gets out of the tumor that way. Those are either called angiogenesis if they escape through the blood vessels, or lymphangiogenesis if they escape through the lymph vessels. Well, when we created our first model of angiogenesis, and we were looking for the genes that turned on and turned off angiogenesis, we had to first find out is, if we created this model, would the model itself screw up the gene expression pathway and cause us a false positive or a false negative? So what we do is we basically take a blood vessel that's a resting blood vessel. We can take it out of your hand. We can take it out of your belly while we're in there. We can take it out from, a lot of moms donate their placentas, and we take the blood vessels out of the placentas that they would otherwise throw away. And we then turn on those blood vessels, get them to grow, and we compare genes from resting to angiogenic, and we look at 38,000, 37,000 genes on an Illuminata gene chip, and we can tell you, A, how turned on or turned off they are, and whether that's statistically significant. This is the human placenta. If you look at it, there are some big blood vessels here. This is the umbilical cord. That's not what we're after. We're after these placenta vessels. We open that up. We have a couple of surgery residents who get a lot of experience dissecting out blood vessels from these placentas. We then take the placental vein, that's a tube, we split it down the middle, lie it out flat, and then we take a skin punch, which is like a paper punch, and we put 2 millimeter punches, and we put those into a blood clot without blood. It's called a fibrin thrombin clot. Hello? No, there. And as you can see, the fibrin thrombin clot, here it is. We overlay it with a liquid media, and this is in a plastic plate that has 96 of these wells on the plate. We use 60 of them. And this is one of those plates. You can see they're 12 across by 8 down, and each of these things, we put a little piece of blood vessel or a piece of your tumor in and go from there. And the guys in the lab call these my chia pets. You can see the tumor. This is a fragment of tumor. Every one of these things here is a blood vessel growing out of your piece of tumor. In the old days, everybody thought that you couldn't culture neuroendocrine tumors because they were too fastidious. We just had to learn first that they had to have this fibrin thrombin clot that acted like a rose trellis that the blood vessels grew on, and once we did that, we were on the way to growing these things. And then we asked 2 things. Out of the 60 wells per plate that we grow, how many of the fragments start to grow? So if half of them grow, your initiation is 50%. Then we ask, if you look at how they grow out from the cut edge of the tumor or the cut edge of the blood vessel, that tells you how much growth there is, and then we put those 2 things together for what's called an overall angiogenic score. So we'll sort of try to show you how that works. So if in the first case, here are the wells. If some of them grow, some of them don't, you just figure out what % grew new blood vessels, and those are the dark ones. You then look under a microscope, you divide that fragment of tumor visually into 4 quadrants, and then you score each quadrant 0 to 4. So if there's no growth in that quadrant, you give it a 0. If you got a whole bunch, you give it a 4. And again, here is early on, maybe day 2, day 3. Here's day 14, a lot of growth. You can see these things branch, they anastomose, and they grow long term. They have been subjected to CD31 stains, factor VIII stains, immunomicroscopy, electron microscopy, and these are real blood vessels. They have Weibel-Piloty bodies, tight junctions. They're the real deal. Again, if you look at how a disc looks under the microscope, that's how it looks to the people who grade them. If you grade it and you grade it and you grade it, and we've done that experiment for validation, you'll be about plus or minus 1. So 0 to 16, you'll give it a 10, you'll give it 11, you'll give it a 9, but it'll be in that very tight sort of range. So what do? We take the tissue before we start, and then we recover it from the fibrin thrombin clot, and the ones that have blood vessel growth, and then we subtract and make RNA, and then ultimately we try to put this on the Illuminata gene chip so that we can tell whether your genes have changed from when they came right from the operating room and into our assay and then allowed to grow and become angiogenic over 2 weeks. Does the assay screw up the gene analysis? Because if the assay changes the gene expression, then we're sort of hosed. So we have to look at that. And the answer is, if you put your blood vessel in and then you look at it 14 days later when it grows, only 22 genes change. If I look at it before I put it in culture and after I put it in culture for, say, 1 day. And then for each placenta that we use, we create gene expressions to see what was the genes, what were the genes that were in your blood vessel when it was asleep, when it was resting, and what are the genes from the same human piece of angiogenic blood vessel that we started with that was resting, what changes, what grows, what comes up. And what's interesting is, look here into this group, if you look at the number of genes that change up or down, placenta 1 was 5,000, 2 was 5,000, 3 was 30, some 4,000, and overall about 5,000 genes change between resting and growing. Now you say, of these genes that were in each of these, how many of them were identical across all of the things? And so what you find is for this, we have about 150 genes that are up or down regulated twofold. Again, 150, 1,000, 1,000, 1,000, 1,500, 1,500. And you ask, of those 1,500 genes, so we went 5,000 genes change, but then when you look at it across everybody at that table, then it drops down to 1,500 genes, and now you say, what if I ask, you can only be considered to be in the column if all of your genes match up. In other words, you all have the same genes that are turning on and turning off. And by that time, it's down to, it's hard to see, about 1,500 genes. And if you look across everybody, you're down to 500 genes. And if you now ask the question, out of those 500 genes that change when a resting blood vessel starts to grow, about 200 are turned on and about 300 are turned off. So we're now down to 500 genes that we can look for out of 38,000 on the gene chip to say, do you have the genes that will turn on blood vessels or turn off blood vessels? And then think about it. If I can tell, if I now know what 500 genes to look for, I can put chemicals, i.e. drugs like Xylota, black raspberry, etc., and say, do you alter the expression of those genes? Well, do tumors at different sites differ at the gene level? Yeah, you may remember I presented to this group a couple of years ago work done by Erica Lindholm in my lab, where she took primary tumor out of the small bowel, lymph node, and liver metastasis, and looked at all kinds of things: CD31, factor VIII, chromogranin, synaptophysin, mitotic index, Ki67, all those kinds of things. And what she said was, your primary tumor doesn't look anything like the liver disease that you have now. Your lymph nodes are way more aggressive than your primary tumor. And all your tumors look alike. So we can treat you with one drug, but your tumor, all 3 of your tumors are different. We need 3 different drugs to treat 3 different locations. And so now the question is, if that's true, if your primary tumor, your lymph nodes, and your liver aren't even related to one another, and they require different therapy, you would believe that they differ at the gene level, and you would be right. And so we looked at gene expression for somatostatin receptor, i.e. what Dr. Lee was talking about today, receptor type 2 and receptor type 5 that show up on Actuarial scans or the DOTA-TOC or the DOTA-TATE scans. And we said, at a gene level, do those things exist, and are they equivalently expressed on your primary tumor, your lymph node, and your liver? Because if you do a gallium scan, if you do an Actuarial scan, your primary may show up, your lymph node you may miss, and your liver may show up. Dr. Vergolini from Austria shows a picture of a patient on an MRI scan with 2 metastases, one that shows up on an Actuarial scan and one that doesn't show up on the Actuarial scan. They change the peptide that's radioactive to radioactive lanreotide and versus octreotide, and it just switches. So one shows up with one drug, the other shows up with the other drug, but neither of them show up with both the drugs, with just one drug. Okay, so again, how did we figure this out? If you look at it, just looking at the tumor doesn't count. You have to take the tumor and look at what genes are in the tumor. You have to look at the small bowel and ask what genes are in the small bowel. And so we subtract the small bowel genes out of the tumor genes, and you end up with just pure tumor genes. In a similar fashion, lymph node, we take the lymph node genes, we take the lymph node tumor genes, we subtract the baseline or the normal from the abnormal, and gives us, should be pure normal or pure tumor genes. And finally, the same kind of idea with the liver. I take a chunk of liver, I look at the gene expression there, I look at the gene expression in your liver tumor, I subtract normal from the tumor, and I should get pure tumor. Now I can compare liver tumor, lymph node tumor, and primary tumor, and I don't care that it came from small bowel or lymph node or liver. I've gotten rid of all those genes, whether they were up or down regulated. And so SSTR, overexpressed in about 70% of specimens, 4 out of 4 livers, 3 out of 4 small bowel, but only 1 out of 4 lymph nodes. SST5 overexpressed in about half of the specimens, 3 out of 4 liver, 2 out of 4 small bowel, 1 out of 4 lymph nodes. What it's telling us is, you know how Eric showed a picture today, Actuarial scan showed some little spots, and then the gallium scan showed, it looked like Machine Gun Kelly had got the guy. Well, that's what this is telling you, is that even if Machine Gun Kelly shows up 5,622 bone mets, the gallium scan may totally, completely miss the lymph nodes, even though it did way better. So we're making progressive changes in how we're scanning, but that still may not get all of them in every case. We may ultimately, and I think everybody in this room at the doctor level will say, we think ultimately we're going to end up with a cocktail of things that we're going to scan with to get 100% expression. But we'll know if we know which genes to look for. This is sort of a hard thing. Let me just explain that if you look over here on the left, the small bowel, the lymph node, and the liver, you can see what was up and down regulated. Look in your book, and you can see that even though it's in one part of your tumor, this is patient 1, 2, 3, 4, this is small bowel, lymph node, and liver, that just because you got it in one place, it don't mean you got it in all the places. And that was for upregulated, this is for downregulated, and that leads us to the question is, now that we know the 500 genes, can I start predicting whether or not you're going to respond to a drug by gene analysis. And this is a gene for a receptor called fibroblast growth factor receptor 3, FGFR3, and that binds FGF, fibroblast growth factor type 9. And there's a drug that just is starting in clinical trial called davitinib, which is a Novartis drug, and davitinib knocks out FGFR3. So why are we interested in this? Because we can now look at the patient's tumor after we take it out at the time of bowel surgery or cytoreductive surgery. We can look for this FGFR3 gene, tell whether it's up or down regulated, then go ahead and treat your tumor in our angiogenesis assay and see whether it works, and then give it to you, and we'll have 3 levels of information: gene expression, my angiogenesis assay, and a clinical trial, which Dr. Lowell Anthony, who will be here this afternoon, is going to talk about, I hope. FGFR3 is upregulated in net tumor tissue, and davitinib blocks the FGFR3. Here is a result like would be shown to you. Here's the control. You can see that about 65% of the wells that we plated started to grow. This is our kills everything control called gallic acid. Interestingly, gallic acid is a food preservative. Kraft Food got the FDA to allow them to put gallic acid in your mayonnaise and your ketchup during the 50s. It is still on the FDA's GRAS list, generally recognized as safe. So gallic acid, the trouble is it'd be the perfect anti-tumor thing, but it doesn't absorb into your body. So anyhow, but it works fine in the laboratory. Again, now you can see davitinib, and you can see the davitinib is in 4 different doses, and we now know what dose to use, and that's 165 millimolar. And it completely wipes out the blood vessel growth entirely. If you look at growth, the red line, again, a dose response, and the overall effect is the blue line, which is the green line and the red line put together, and you can see that nothing happens, nothing grows in the face of the high dose davitinib. And those are levels that you can achieve in your blood. How do we know? Novartis did a trial where they brought people in, they gave them the drug, and they checked their blood levels of davitinib. So let's look in my assay. We operate on a lot of folks. This is about 100 and some people. The black line is your tumor growing. How much did it grow? This is initiation here with no drug. Now you add the davitinib, and you can see that there are a couple of people who I wouldn't treat with davitinib based on this slide. These down here, I would. Interestingly enough, this black line and this blue line, the slope of those lines are the predictor of what percent of people will respond, and it's in the 90 percentile range that you would respond, everybody would respond to davitinib, and you'd have 3 mistakes. And this is the growth. You can see again in our assay, the growth works like a charm. Davitinib works like a charm, but there are still some people that I might not treat with davitinib until I look at the overall effect. And the overall effect, we have, again, over 95% response rate. This is your control, this is your treated, and there's just a couple of patients up here that look like they're not good candidates. Now, what we need, the next step is to take this data and to put people on davitinib to see if they're going to respond. Well, what does the future hold? I think we will have development of tests that will determine your prognosis. We're going to be able to look for things that are triggers of proliferation, triggers of blood vessel growth, etc. We're going to be able to look for receptors. We got FGFR3, fibroblast growth factor receptor type 3, i.e. put on a radioactive tag onto FGF9 and use it as a scan. We're going to be able to look at your genes in your primary, your lymph node, your liver, and say, are you a good candidate for gallium scan? Are you a good candidate for PRRT at the gene level, not at the far grosser level? Stuff that we can do in the lab for $700, the scan costs $5,000, $2,000, who knows how many $1,000. And we think we can develop non-toxic therapies based on your tumor and blood vessel characteristics. One of the things that you may or may not have run into amongst your support groups are people who have what are called unknown primaries. They come in with tumors in their liver, but we don't know where they came from. And so we can take that liver biopsy, send it off to California to Biotheragnostics, and they can tell us with incredible accuracy where the tumor arose. Did your tumor come from the small bowel? Did it come from the stomach? Did it come from the duodenum? All those people who for years and years said, I don't know where my primary is, we can now have a way to focus in that, all based on the expression of 92 genes. So you're starting to hear numbers that we start out with 38,000 genes, we get down to 5,000 genes, we get down to 1,500 genes, we're down to 500 genes, 92 genes here, and I'll talk a little bit about 28 genes to figure out how black raspberry works. 92 gene assay accurately predicts the tumor subtype. In our cases, we looked at this. There were 30 cases, 23 out of 30. It nailed it deader than a snail. Of the people who we took to the operating room, 100% accurate. And if it says we're confident and we look and we find it and take you to the OR, 100% right. You know, a lot of people write to me, a lot of people talk to me, a lot of people call me, and they say, you know, I read the old data, and trust me, some of the major drug companies publish data that's 15, 20 years old. Well, what are the current survival statistics for people who are treated with aggressive surgery, PRRT, Afinitor, Sutent, all these kind of things in the modern era compared to the old days? And this looks at the NANETS group, what they published in their guidelines, and they said, if you have just lymph nodes, and there were, in our group, 68 people here, their survival was 35% at 10 years. If you look at the more modern stuff that, excuse me, I'm sorry, the SEER data was 46%. Our New Orleans group is over 80%, a 35% difference. And if you now look at people who present to us with liver involvement or distant disease, 10-year survival rate is 63%. The NANETS group says it's only 30%. So basically, we're doubling your survival in the modern era. That, I think, is really, really important, that you don't become depressed looking at the old NANETS data and the SEER data because it's ancient and old. This is just graphically to show you how the lines look. And as you can see, the group in New Orleans, the line is red. versus the green line, which is the SEER data. That was 2 years ago. We're now even slightly better at 5 years. You can see at 10 years, we're now at 77%. And now we're able, through what's called Kaplan-Meier, to predict that basically 40% of you are going to be alive, even if you had distant disease when you walked in the door 20 years after we saw you for the first time. That's a huge change in what's going on. For people, it's like asking questions, and I'm going to show you very unique stuff done by a young lady who was our administrative assistant now in nursing school, named Ann Diebold. Rather than looking at survival, she asked the question, if you were an insurance person, you would create an actuarial table to tell you how you're going to survive if you didn't have anything. If I didn't know who you were, that you're a 35-year-old woman and you're white and you're blah, blah, blah. Okay, so, and this is that survival. And this is, if you didn't have any disease at all, you were the green line. And if you had distant disease, mid-gut carcinoid, and you came to us and we did our aggressive therapy, you can now look in the far column, and you can see that at 5 years, there's a 13% difference in survival. So at 10 years, it's 22%, and at 20 years, it's 35%. So what does this say? Is that we've now not only made progress in increasing your survival, but the difference in you having and not having this disease is now down in the 10 to 20% range, i.e., go about your life, live a normal life. Don't do the ostrich thing. Don't be scared about aggressive therapy, because you're going to live damn near a normal life expectancy. And again, pNETs, I think we're about running out of time. But again, the SEER data says that 7% of you are going to be alive at 10 years. If you were aggressively treated at our institution, half of you are alive, a difference of 42%. So don't look at this old data and say, I'm gonna die, because it ain't true. You have to look at this as that old data is old, and you need to get up-to-date modern data and base your life and what you're gonna do and you're going forward on new data that says, boy, you're really in pretty good shape. Thank you very much.